And of course, I'm going to shut my sound off. We're live with Otangelo, and Immutable is going to be sitting in the back room monitoring us so that we behave. And
yeah, I put the behave part in quotations.
Yeah. Yep. Well, you're good hall monitor.
Why don't I try?
I'm surprised at how long it's taking YouTube to. Oh, okay. YouTube is finally live. So I shut my sound off. Anyway, so what I wanted to go over, and I have to share a screen, is this one comment that you came up with. And this is about the leap of logic, but I'd like to have you tell me a little bit more about it. Let's see. Android immutable. Okay, Evo Google Docs. Share. Okay. So this one, is that big enough for everybody to see? So can you see it?
Yeah.
Okay. So I broke it up sort of I like to break things up into bullet points. So anyway, so first of all, you said, do you want to, let's see, after all, to see your worldview without God confirmed? Or I think you were saying that that's what you think I want to do, is to see my worldview without God confirmed?
Well, I think that we all have a bias. I mean, that is not, we cannot deny that. And I have bias, of course. I want to see what I want to be true, to be confirmed. And if you value your worldview and you confirmed and confessed that you're an anti-theist, so of course, if you want that worldview to be true, then I think that you want to have it validated by the evidence which we see around us. That is my supposition, at least.
Okay, that doesn't characterize me at all. I don't care if my worldview is confirmed. I believe in my worldview. If evidence were presented that changed it, then I would just change my worldview. But being an anti-theist, that's more of a political position. That's not a worldview. Being an atheist, it may be a worldview, or being a naturalist is a worldview, and that's how I think things work. But let's see. I think evolution is demonstrated with all of science, so I don't see a problem with. I don't think there's ever going to be a moment where we say, "Aha, here we have proven it." But I mean, we've proven it enough with the little tiny things. So anyway, but I just wanted to point that out, that no, my worldview doesn't enter into it. When I study science, I'm not thinking about my worldview.
Okay.
I don't usually care.
Yeah, I mean, I don't put all atheists into a bucket. We know that there are all kinds of flavors, the same sense as there are all kinds of flavors of theists. So, but I mean, I want my worldview to be true, and I don't deny that. And of course, if the evidence which I see around confirms it, then I'm intellectually satisfied.
Yeah, I don't think I have any need for my worldview to be true. It doesn't matter to me. Whichever way the wind blows is the way that I would go. So I'm not really out there trying to sail to a particular island. I'm just, whichever way the wind blows, I will go. So anyway, so I don't really care. Let's see. I do like the theory of evolution, and I think it's beautiful. The whole story of natural development. So I mean, I am a little attached to that. Probably, to the same amount I'm attached to maybe the Star Wars series or something like that. Okay.
Well, you like science fiction.
I do, yeah.
And I regard primary speciation and common ancestry as science fiction based. So.
Yeah, I know. Let's see. Then you say, you, an atheist, on the other hand, confronted with the scientific facts, must do unbelievable mental gymnastics to put the dots together. I don't think that's true.
Oh, I think so. Absolutely.
Mental gymnastics. I don't think we do any. I think leaps of logic would be mental gymnastics, but I don't think we do mental gymnastics.
Yeah, that is the entire dispute and disagreement between us and between us as believers that there must be a divine, super intelligent agency behind what we observe in nature, and on the other hand, you and unbelievers, which think that natural, non-intelligent mechanisms suffice to explain what we see in nature. That is the entire dispute here, which is something which never ends.
Right. And you say, but I commonly suspect that you don't know how to make a logical leap, like code comes always from a mind. Okay, tell me what a logical leap is.
Well, I think it is very obvious, Speed, that when you have to make an assignment of meaning, that this is only something which intelligence can do. You have never seen a translation book from English to Chinese, for example, emerging by non-intelligent action and mechanisms. And in order for you to believe that the assignment, the genetic code and the assignment of codons to amino acids can emerge by unguided events, this is a logic leap which I cannot follow, and I cannot follow how you can actually even believe that this is possible, because conceptually it simply makes no sense, because matter has no urge to assign anything. It is not intelligent, and I cannot understand how you can even accept or think that this is possible. This is something which doesn't go into my mind. And John Maddox has made recently a video on this sarcastically and making fun of atheists. It's not my style, but I can fully follow why he ridicules atheists, because it simply makes no sense.
Well, John Maddox isn't very bright, so that's why he ridicules us. But you say, but I commonly suspect that you don't. You're telling me that I can't make the logical leap, so that implies that you have made a logical leap, and I think you're right. But logic, okay.
I don't even think that. I don't even understand how, as an intellectual exercise, I need to make any kind of effort to understand that, because it is so obvious.
Right. It's an intuition. Okay. If you've studied logic, you'll know that they never talk about logical leaps. There is no logical leap operator. There's only and, or, and, and not. So, and implication, there's no logical leap operator. So what we have here is that you have a strong intuition. Now, you said that we have never observed a factory or a translation being done without an intelligent mind. We have never observed a cell being created with an intelligent mind. So I don't understand. So this has to be intuition, because you have no evidence of an intelligent designer. You've never seen this designer design life, just like I have never seen a factory pop into existence because of a landslide.
But for these side chats, I have a whole list of sentences which are less than 200 words long, and I can just read one which is quite short and brings it straight forward to the point. Gravity is inferred by observing an apple falling to the floor, so the existence of a non-physical, non-created creator is inferred by observing the existence of a finite universe. We do not need to experience God by our senses, and it doesn't require that we can see him or that he demonstrates himself to us in order that we can make that logical leap to the fact that creation points to a creator.
That sounds good, but okay. So we infer gravity. I take a ball, I hold it, and then I drop it, and it falls to the floor. And then I can measure that, and I can take another ball, and I can drop it, and it falls to the floor. So that happens every time. But I've never seen an intelligence design life. So how do you confirm that by observation?
Because, I am making this argument, Speed, for months. And people, they are even actually a little bit saturated and bored because I am repetitive. But it is basically what I am saying all along, and I can repeat myself again. Intelligence makes instructional prescribed blueprints, and upon these, then specific devices are made for specific purposes, and that is analogous to the genetic code which is used to generate information stored in the genome and in epigenetic languages, which directs and controls the making and operation of molecular machines and interlinked cell factories. And that is analogous to what we see. So we can make a logical leap because we never observe that random unguided events are making these things. They are making a language and information and then direct the make of machines and factories. That has never been observed. And what we see is that science is trying in the last 70 years to come up with a natural explanation, and is miserably failing. Rather than coming closer to close the gap, it is becoming bigger and bigger because the more science investigates molecular biology, more and more layers of complexity come to light. And that doesn't help materialism. It plays into the hands of those which propose a divine agency, which made everything.
The more you study a rock, the more complicated it gets. But okay, we have not observed a natural system create life out of basically non-biotic elements. We have observed a cell, a single cell, just a little bag of proteins. We have observed that create a human being, or create a chicken or a fish. So we have seen that. So we've seen some pretty magical things happen all through natural processes. Okay, that's number one.
But the zygote has already all its information stored in the genome and in the epigenetic language. That is already there. And I mean, abiogenesis starts from the lack of energy, which you need the matter, the basic building blocks, and the information.
I think it was Yaki or somebody like him who pointed out that you can take two identical eggs with the same information and they never turn out exactly the same way. They turn out sort of the same way, but not exactly. I have identical twins. They didn't turn out the same way. So it isn't just the DNA, and they have identical DNA, but they have a lot of differences. So there's more.
That's what I am saying. Yes, we agree. That's what I am saying, that epigenetic information is also strongly in play. I mean, you have the entire gene regulatory network. You have all these mechanisms which I am listing in my write-ups, which I have investigated, and you know what I am proposing, that signaling pathways, epigenetic information, and genetic information is actually the source to explain organismal complexity and body form.
Yes, complexity. Like I say, rocks are complex too. But anyway, you do this, or assignments must be established by intelligent rules require a rule maker. These are all assertions. You have not shown that to be the case. That is always the case. Now, when you make this logical leap, like I say, we've never observed an intelligence design life, ever. Not in the lab, not us, not humans, not anybody. We've never observed this. And you're saying we've never observed a natural system create life, right? Because we know that it happened 4 billion years ago. And we have never observed God make life because we know that happened 4 billion years ago. Or in your case, what, 6,000 years ago? So well, you know what happened in the past, but you haven't observed it either. So how do you know?
Yes, that's why we use abductive reasoning to the best explanation, and eliminative induction. When you have two options and one doesn't work, then the other by default is true.
So how do you abductively arrive at God?
I just gave you an example, Speed. Show me a natural mechanism which can make codes and factories.
Show me a God that makes life. Show me a God that makes codes. Show me a God that makes proteins.
That is that is the logical inference and abductive reasoning to the best explanation, that intelligence, a divine
How do you know it's the best
creator,
one?
well, because we know what random mechanisms, what the range of natural mechanisms is to produce such things. And we have simply never observed that non-intelligent mechanisms are capable of doing all these things. And
But we've
I mean,
observed
science is,
non-intelligent mechanisms turning zygotes into humans, and that is quite a stretch. That is
well,
magical.
but you have already all the information to have the development of an organism there in the zygote. So you have to ask yourself, where did that information come from?
Right. Where did it come from? That is the question. So why would why would God be a better explanation than X, or we don't know where the information came from?
Again, because we know by experience that intelligence can do all these things.
Oh
And
no,
we
but
know
they can't, because we have never created life. So we can't create something as complicated as a cell. We can create machines, but we have never created a cell.
Okay.
So how do we know that?
Okay, so what we make with all the intelligence that we have at hand, Mike, is far less sophisticated than what we see in biochemistry, which is far, far more sophisticated, going up to the limit of what is physically possible. So what is the logic then? Is it logic to say, if we see a system which is even more complex than those which we as intelligent beings can make, that then there must be a far more intelligent divine designer behind these things? Or shall we say absolutely no intelligence is a better and more capable mechanism and explanation for what the evidence is in the natural world?
We don't need any intelligence to turn that little string of nucleotides into a human being. So why do you assume we need an intelligence to create that little string of nucleotides?
But Mike, I just told you
that it can happen naturally.
But Mike, I just told you that the information in the zygote is already there. So of course, if it's already there, then it is possible. But how do you get that?
But if you take DNA and you put it in a test tube, nothing happens.
Yeah, exactly. That's my point. That's my point. If you if you take all the ingredients, take a living cell, and then you take all the ingredients and put it into a test tube, and then you scramble it for 1 billion years, you will not get life.
Right.
So that is my point.
You have to have a living cell to create a living cell. But where did the living cell come from?
Life.
So you don't know where the living cell came from, and you're assuming it is some magic being who is greater, who is like an ape. He's like an ape because he's intelligent like an ape. The only intelligence we have ever observed create anything are human beings.
But why do we have to?
No raccoon has created a factory, right?
But Mike, why do we have to observe it? Why can we not make a logical leap and a sound logical inference to the best explanation? Because we have not observed abiogenesis and a primary speciation.
Okay, but your logical leap is far greater than mine.
No.
The only logical leap that I make is that there's a strand of DNA with a certain sequence of nucleotides in it. Those nucleotides change a little bit, they mutate, and they create now we have new information, and we create a new life form.
You do believe. You're not starting from scratch, Mike. Where do you get your nucleotides from?
We could go back to that, but we're not going to. We're not going back to that. We're going to start with nucleotides. If you have a string of nucleotides in a test tube, it does nothing.
Yeah,
but if you have a string of nucleotides in a living cell, it does something. And we can show you how that living cell from one organism to the next changes just a little bit. You do believe that, right?
Well, I told you what I believe in regards of how all complex organisms are built. and and it's not just genetic information. It is also signaling. It is epigenetic information, and it is
Wait, what's epigenetic information? Where does that come from?
Yeah, that is a good question. Where does it come from? Where does information in a general sense come from? And where does the language upon which the information is stored come from? It's not about just the information itself. It is also about the language. It is also about the storage devices which you require to store the information. And it is also about the information transmission systems, like we know from genes to amino acids. All this has to be there at once. If you don't have one of these four things, you have nothing.
Okay, you said epigenetic information. Where does that come from in the cell? We know where the DNA comes from. Where does the epigenetic information come from?
Well, we know only about maybe 2 or 3 storage mechanisms of epigenetic information. One is the histone tail, okay? Then we have also the glycan code, which is stored in the glycans of glycoproteins at the surface of the cell, at the membrane. And the structure is semantophoretic. It stores information. But there are about, I think, 29 different epigenetic codes, which I am listing, which science doesn't know where that information is stored.
Yes, it's stored in the DNA.
No, it's not.
Oh, but it is. That's where the epigenetic modifications are from, proteins and enzymes, and those are stored in the DNA.
Okay, partially I agree with you, of course, but
mostly.
It's not, well, science doesn't have a clue, for example, where the splicing code is stored. It's not in the genes.
The splicing code is in the genes. That's how genes are spliced together.
No, it's not in the genes.
Okay, where is it?
What science doesn't know.
We know how splicing happens.
That's not the question, Mike. It's not how it happens. It is how does the cell know how to, for example, the human genome has about 20,000 genes, and the human, and we have about 6 million protein species. And that difference, that means that one gene can be spliced about 300 different ways. And always you have a functional protein species. And the question, of course, is how does the cell know where to splice in order to make a functional protein product? And I've asked Sternberg about that. Where is the splicing code? That information about how the cell knows where to splice the gene stored. Where is that information stored? And he said, we don't know. Science doesn't know.
Who did you ask? Who did you ask?
Sternberg.
Who's Sternberg?
Well, I don't know if you remember that in 2004, Steve Meyer, he published a book about genetic information, and he was the reviewer. And he was, I think, at the Smithsonian Institute, the museum. He was a curator back then, and he also was making peer review for the Washington Journal, something like that, actually. And that paper of Meyer went through. He accepted it. It went peer review, and that was a big outrage.
Yeah, he had to resign.
Yes.
Yeah, because it never should have made peer review.
Yeah, but that's another topic. Anyway.
Okay.
I talked with him about this, and he said, actually, we don't know where. Well,
he doesn't know, but I know it's in the DNA. That's where the splicing information comes from. That's how exons. How do you think exons get spliced out? There's signals in the introns. You know that, right?
No, if you have a science paper on that, then I would like to.
A science paper on introns? I have.
No. What I want to know, if the information for the cell to know where the splicing has to occur, if that information is stored in the genome. If it is so, okay. I mean, I don't know it, but if you have a science paper which confirms that, then it
No,
adds up to my knowledge in regards of that issue.
I'll have to search ancient history for that because it's in the textbook. If you look at molecular cell biology, a really old textbook, from around 2000 or the late 90s, you'll find that information there about how splicing happens. It's in the genome. The signals are in the introns.
Yeah, the question is not how it happens. The question is, where does the information come from?
Well, the same place
for the spliceosome to know where to splice, actually. That's the question.
It's in the genome. It's in the genome.
Okay. I mean, if it is in the genome, I'm fine with that. But, I mean, I am not obliged to know everything. So that's something which I'm not knowing.
Okay,
and
I'm telling you, we know how that works.
Okay, that doesn't mean but that doesn't mean that all the other epigenetic languages are also stored in the genome. As I said,
also stored in the genome. It has to be, because the only thing that happens in the cell happens from proteins and a lot of the information comes from the environment, the signals, right? But the biological information comes from the zygote, from the mother. So one cell passes down to the next, and it gets mutated over time. And that's how that is the proof of evolution. That's how it works. That's all you need to know.
Well, the thing, Mike, is I gave you two epigenetic codes, which we know that they are not stored in the genome, which is the sugar code and the histone code. These two codes, they are not.
Code? Wait, okay. Tell me about the sugar code. How does that work?
That's the glycan code, which is the proteins. They are glycosylated, and that glycosylation is information bearing. And these proteins, they are membrane proteins on the surface of cells, and they are communicating with neighboring cells, and they bear information. Now,
that glycosylation happens with enzymes and proteins, and that information comes from the DNA. So all of the glycan code is in the cell.
Well, the fact
that it comes from the mother.
Well, Mike, I disagree with you, because the glycan code is actually
wrong.
The glycan code is far greater and stores much more information than genes do. So,
do you have a paper? Do you have a paper that states that?
Yes, I have an entire article. I can put it here in the link, and then you guys can look it up. Just a moment, please.
Okay, but it's wrong, or the way you're reading it is wrong, because the glycosylation happens with enzymes.
Just a moment. Just a moment here.
You're reading articles and you're coming up with a layman's opinion about them, and that does not explain fully what the glycan code is. The glycan code is something that we labeled a whole bunch of different systems as codes, but they all come from the DNA.
Well, I disagree with you, Mike, because the histone code is stored in the tails of histones and not in the genome.
Could you put these things in?
I'll put it here in the public chat. Just give me a minute. I've put it in the private. I'll put it here in the public chat too.
We're probably going to have to defer this, and I will comment on it later. But no, the glycan code, you're wrong about the glycan code. About as wrong as you could be.
Well,
there is no secret information in the cell other than what comes from the mother, from the zygote. There's a whole lot of mRNAs in the mother that basically get transferred down. And there's also cell membranes, and there are some existing proteins, and all of those things are eventually recreated from the DNA and in that cycle.
Okay, here I'm reading. The majority of eukaryotic proteins are modified by co-translational and post-translational attachment
Of course they are.
of complex oligosaccharides, glycans, to generate the most complex epiproteomic modification, protein glycosylation. Okay, and
Okay, what are you reading from?
from the link which I gave you. If you open it, then the second thing written in red is where I'm reading from.
Okay, what is the source of that? What is it? Did you
Source-
write that?
No, I can give you the source. Just a quick minute. Just Googling it, and then I'll give you the source.
But we're probably going to have to move on.
Okay, I have it here. I can give you here the link. Yeah, I mean, you can have a look. It is a very interesting topic, and then we can have another chat eventually if it is of your interest. Okay.
Yeah, I can explain how you're wrong about this.
Okay. If you think I'm wrong, here is the science paper. I'll put it in the public chat as well. Okay.
Let's see here. The science paper that you're reading from, why am I not seeing it? Oh, you put that in private chat?
I put it in the public chat as well, in both.
Why did I not see it? Oh, Smokey is not blocked? Oh my God, Smokey's not blocked. That could be catastrophic. This channel famously blocks Smokey. I can, hopefully I can get this tab up. Okay, glycosylation in sugarcane. Okay, that's interesting. Okay, anyway, moving on.
Okay.
Let's see. So right here we have we have logic gates and we have a complex. We're looking at the mTOR pathway. It's involved in insulin and it's involved in cell growth and cell apoptosis, and it has a number of different inputs. Different types of signaling, and the pathway, as you can see, is very complex.
Yes,
So,
I did write a paper on that mTOR protein, which is a central hub of signaling in the cell. It's amazing.
right, you see these question marks here?
No.
Where my cursor is? I don't know if you can see where my cursor is.
No, I can't.
You can't see my cursor. Oh, now you can see my cursor. No? You can't see my cursor. It's too bad. Anyway, there are a number of question marks in this pathway. This is a very, very complicated pathway. Some of those question marks are things that we don't know yet. Some of them may not actually do anything at all. We know they have.
I have an article on that very intelligent protein, mTOR. I'll put it here in the private and public chat.
Okay, so is this how you would define complexity in the cell?
That what we are seeing here is hypercomplex, of course.
Okay, hypercomplex. But now you'll notice if you follow one of these pathways, let me see if I can find one here. Like down on the bottom, there's S6K1, which turns off EF2K, which turns off EF2. And you can see that pathway is overcomplex. That's all S61 needed to do was to turn on EF2. So you can see that there are useless steps in this pathway.
Well,
and
Mike,
these are the logic gates you're talking about.
I doubt that science has fully figured out how all this complexity.
Oh, so you're going to say, oh well, we don't know, right? Well,
yes, I am saying it is very well possible that science has not yet fully figured out how that kind of complexity actually works. And like with junk DNA, in the future they will say, oh, okay, what we thought is useless has actually functioned. I am very strongly thinking that this will happen.
Okay, you're basically making another logical leap that we're going to figure this out, and we're going to figure out that everything. That there's a reason for every one of these steps, but that is just your appeal to your worldview, to your belief that there was an intelligent God that did all of this. And I can say the same thing. I can say, we don't know exactly how abiogenesis happened, but we're going to figure it out someday. So what's the difference between you saying we're going to figure it out and me saying we're going to figure it out?
Okay, the difference, Mike, is that We have with abiogenesis a conceptual problem. And that problem is that molecules, they disintegrate, they do not complexify, and molecules, they do not make languages, they do not make codes, they do not make assignment of meaning. This is something which only intelligence is known to be able to do. And in regards of signaling here, this is basically an argument from bad design, which I am saying is that science doesn't even know the language of signaling, of many signaling pathways. And this is something which science is very at the beginning yet. And as Salvador said, in the next 10 years, they will make huge advance in understanding how that kind of organization and complexity actually works. And what they will unravel, everything indicates that it will be awe-inspiring, and that what today is still not known will be understood. And I believe that it will be more and more evidence of design.
So now you're making an assertion that we're going to find out that God did it in the next 10 years.
Well, I believe that what science will unravel in regards of more and more levels of complexity will more and more evidence divine design.
You believe?
Yeah, I believe.
Yes, that's all you got. You can't prove it. You believe it.
No, I don't. Well, that's a faith statement, a prediction which I make in regards of intelligent design.
Well, I predict that we aren't going to cure all these question marks in this mTOR pathway ever. There are going to be some. There are going to be some of these pathways are just random mutations that are neutral. That's my prediction. So I believe, and you believe.
Well, that's possible that a part eventually is already a product of entropy in the cell. That's possible as well. Since I do not know in depth what we are talking about right now, Mike, I can only give a guess and an opinion which is not informed. I have no basis to say it. I'm just confessing what I believe.
Yeah, and that's kind of what it's down to. You believe and I believe, and you said, okay, you think we're going to find out, and I think we're going to find out that abiogenesis basically has perfectly good natural pathways. You think we're going to find out that that all of these things were done by God? I mean, look at this diagram. Would you hire an engineer that built something like this?
Well, first I would have to understand it to then give you the answer to
there's a whole lot of these cases where you have two NOT gates in a row, which is just a pass through. So NOT means it's true, right? It's a one.
Mike, you're basically trying to make an argument of bad design, and that is an argument which
I'm making an argument from design. Okay, I'm looking at the design.
From bad design.
Design is bad.
Yeah, exactly. You are making an argument of bad design, implying that it is not designed. But I mean, I always say, if you see a cell phone which has a problem with the I don't know how you say in English. If you see any defect on your on your cell phone, will you also say it was not designed? Of course not. You will say there is some defect on it, but you would not make the leap of a conclusion that it was not designed. And the same thing is with nature. I mean, you eventually detect something which you think, oh, that is not perfectly designed, or it's faulty or so. I mean, a classic example is the laryngeal nerve in the giraffe that has always been brought up. And there are other few examples like the appendix and so forth. But these are really worn out arguments against design, Mike.
Okay, so follow me here, Otangelo. So you're saying that some of these pathways, some of these lines that go through that end up with question marks, you're saying that some of these are due to a defect.
Maybe as I told you, I don't understand the diagram.
Okay. But
so
you did say that. You did say that. You said that some of these are because of genetic entropy, because of a defect, right?
It can be. It
can
be. Maybe.
So do you realize what you just said? You just said that pathways can be created by random mutations. You just admitted that pathways can be created by random mutations.
No, I didn't say that, Mike.
Oh, but you did.
Okay,
you suggested that some of these things that don't make sense.
So I didn't express
mutations.
myself well, Mike. That's not what my
Well, you were
intent
talking
was.
about a defect in a cell phone. So if you have a defect in a cell phone, there's obviously something wrong with it, and we're going to say, oh, well, it wasn't designed. But the defect in this particular cell phone that we're looking at in this mTOR pathway, some of these defects have created pathways. These are all valid pathways. Some of them indeed, and you are correct, some of them indeed are mutations that ended up doing nothing, but wasting a little bit of energy. But you can see that pathways can develop by mutations.
Like nobody is
information can be added.
Mike, nobody is denying that mutations happen. Nobody is denying that mutations, they can have a detrimental effect, and very, very rarely they can even have an advantage for the organism. This is also known. So what is your point, Mike?
My point is that random mutations can create these pathways. With a system this complicated, very small mutations in these genes and in other genes or these particular proteins and other proteins can result in different pathways where one thing will turn another thing on. If you look at the logic of this pathway, this is the logic of a child. You have one thing turn another thing on. Sometimes you have two things two different ways that something can get turned on. Sometimes you need two different things in order to turn something on. So we're talking about and/or and not gates. And you can see that small modifications will allow these things to fit together in different ways, and we can create pathways by modification. And that's all evolution is telling you.
Well, I mean, making the leap of faith to claim that
No leap of faith, you can see it in the chemistry. I can show you the chemistry.
Well, you can show me the chemistry, but you cannot show me that all biodiversity came by these mistakes and by these genetic
I can't show you that all of anything does anything, but I can show you the specifics of how each individual part works. I cannot show you how an entire factory was created, but I may be able to take a picture of a construction guy hauling a log or hauling a beam or welding a joint. Okay, I can't do all of anything, and that's asking too much. If I show you each particular part, and there is no logical problem with creating a new pathway through mutations, then there is no logical problem with the entirety of the development of life from a single type of eukaryotic cell.
Okay, Mike, first what you need is a signaling language. You need epigenetic.
No, you don't. Okay, we have a language right here, but the language is just a bunch of proteins clicking together and magnetically holding each other. This is the language. You tell me how this turns into a language. Please turn this into a language. Define this complexity.
Well,
as a language.
As I told you that the Boolean logic used in gene regulatory networks, there is a logic implied which we are as humans applying in electric circuits. And electric circuits, they do not emerge randomly by unguided evidence. Because
electric circuits aren't life. We're looking at life. We're not talking about electric circuits.
Yes, but gene regulatory networks, they use exactly the same logic, which is Boolean logic, which we use to make electric circuits and computer boards. So how do you explain that?
Look, RAS is okay at the top of the diagram. RAS is phosphorylated. Okay, it turns on RAF. It turns on MEC. It turns on ERK1 and 2, which turns on RISC, which goes down and inhibits TSC2. Okay, up here we have frizzled, goes down to DISH, turns off GSK, which turns on TSC2. Okay, what programmer would design a circuit to turn for one thing, to flip one bit, to flip another bit, to flip another bit, to flip another one on? Why not just go from RAS straight down to TS2 and inhibit it? What kind of intelligence is this that you're looking at? You're looking at complexity. You're not looking at intelligence.
Well, first of all, we know that whatever we see here, it works because we are here.
Right. It works because we're here. Well,
Okay.
it works well enough for us to be here. It
So-
doesn't. It isn't necessarily efficient, perfectly efficient. A programmer could have done a much better job of this system.
Well, the question is, efficient or not, can unguided random events create logic gates? And
Well, we've
Boolean
already
logic
proved that it
events?
could. We've already proved that it could because
You show that it is possible? I don't
because
think
RAF
so.
turns on RAF. Okay, so if we have some connection where basically RAS enables RAF, and we can show you that in chemistry how that can happen, and we can show you that random mutations can change these things from one system to another. So we've shown you all the steps you need to develop this naturally. We've shown it to you, but you cannot make and I wouldn't call it a leap. You're not making a proper inference because you're not looking at how this could have developed. You're only looking at how God did it because you are so stuck in your belief, Otangelo, and I can't shake you loose from it no matter what. I can show you each individual part, but you are unshakable in your belief. And that's because you prior believe in God, and that is getting into your science mind. Now, I don't prior believe in evolution when I learn how these gene networks work. I don't need to. All I need to do is to look at the details. So I'm showing you the details, and I'm trying to show you that we have shown you a sufficient number of details to show you how evolution actually works, how it happens.
You are showing me a diagram which I don't understand, which probably you also do not understand. And based on that, you are trying to convince me of something which I cannot be convinced if I don't understand what you're talking about.
I understand some of these pathways, and I understand how they work, and I know it's chemistry.
But I don't. Yes, but I don't. And so you are trying to tell me something which I cannot give you an answer because I don't understand what you're talking about. But
okay,
we can go to the core of the gene regulatory network, and you remember that some people have claimed that there is actually no logic employed in the gene regulatory network, and that is something that we wanted to talk about. And I've made a little investigation in regards of that. I put a link in the private chat, in the public chat, and what I have investigated actually confirms what I am saying. And
it confirms your bias is all it does. Because you keep applying your bias, but you're not listening to me when I tell you how each individual step in this pathway can be created by mutating DNA.
Well, shall I simply believe what you are saying I'm not simply believing what others say.
Well, no. What you do is you go and you investigate and learn how protein changes, how one protein or enzyme or one of these signaling molecules can change into another one. There's information out there in chemistry books, in biology books, in molecular biology books that can tell you exactly how you get from one protein to another through duplication with modification or through de novo gene production or whatever, through splicing, a slight change in that splicing information, which is in those introns.
Nobody possible. Nobody doubts that it's possible. But if we are going to the bottom and to the core of the issues, and you know that because we have
it's the core.
No. The initial steps have to be elucidated, and as I said previously, there are conceptual problems with naturalism and materialism because matter does not select things. It has no urge to select.
Avalanches happen, like one rock will hit another rock and move that rock. That's the same with chemistry. It's no different. It's just a little bit smaller.
Well, you have to make a selection of 20 amino acids amongst 500. You have to make the selection of ribose amongst many different sugar types, and the one which is used is the most unstable one. You have to, science has absolutely no clue.
So now you're moving the goalpost back to abiogenesis.
Well, but you want to start at the middle of the journey, Mike. And if you would understand,
that's where you're starting. That's where you're starting. You're saying how did today's ribosome develop? That's the middle of the journey. You got to pick a spot. And what I'm showing you here is a very small spot, something that we know a lot about, the mTOR pathway. And I'm telling you that every single piece of this has a paper behind it that can show you exactly what these genes are, and we can investigate each one of them and we can look back and we can look at other organisms that are extant, and we can figure out how these genes have been modified over time between organisms. And that is what evolution is. That's what evolution does. That's what biologists do in order to prove this. So
you will arrive at
hang on a second, I'm not done yet. Not done yet.
Sorry,
if you dip down far enough and you look at the minutiae, if you look at the details of how a zygote develops into a fully formed human being, you're going to find all of these mechanisms that you're asking questions about, and you're going to get to know them. And when you get to know them, then you will have a clear intuition about how this stuff developed naturally. And I do. I have a very clear intuition about how this stuff developed naturally that is backed by a whole lot of very detailed knowledge about each step of this process. And that's what you're not doing. You're stopping. You're coming to a full stop when you see the complexity, and you are not drilling down into how the complexity could have happened. Go ahead.
That's what I do all the time, Mike. I mean, I always like to go to the bottom of the issues, and our intention was today to have a look if actually the gene regulatory network uses logic gates. That was the topic tonight, and as I told you, I'm prepared to respond in regards of that. I'm not prepared to respond in regards of that diagram because I have no clue what you are showing to me, and so I cannot give you any answer whatsoever because you are telling me something which I don't know anything about.
Okay, the most okay with abductive reasoning, the most satisfying conclusion I can come to looking at this diagram is that the spaghetti monster created this spaghetti code. Okay. Okay. I think.
Okay,
I made you laugh. We're coming up on an hour, Otangelo, and we're probably just going to spin around each other. But I would like to. I guess if I were to approach this, I would pick one of these pathways, and I would start looking into every protein and every step, and come back at you with more details. That's how I would approach this. But I think
I'm open to do that, Mike. But what we need to do is always to go to the bottom of the issue and see here really what is at the bottom to have an understanding of the thing. And then we can say something. And I think in regards, I come back, that was our topic tonight, in regards of gene regulatory networks. What regulates them? They are working based on Boolean logic, which is analogous to what we use to make electric circuits. And you have to ask yourself, how can natural mechanisms come up with Boolean logic? Explain it to me, please.
We're showing it to you right here. If you create, if these proteins somehow are created through duplication, mutation, whatever, de novo, gene production, various variable intron slicing, this is the logic gate I'm showing you. Look at TSC2 in the upper 2/3 of the screen. It's a blue thing with a bunch of little P's above it. Okay, that has a bunch of knots on it. Like it has knots, it has 4 knots, and it has 2 activators basically. Okay, and this thing is of a particular shape where it fits with TSC1. So this is how it works. This is the logic gate that you're talking about. There is no electronic logic gate that I have ever seen that looks like that. I could write it. I could do a circuit like that, but then of course this doesn't work like a circuit anyway. But anyway, I'm getting tired, Otangelo.
Okay.
I think we kind of let it go. This the mTOR C2 pathway is something that very much looks like it was duplicated and created later than the mTOR C1 pathway. Which is very interesting. But this is a really interesting set of pathways, and it's been well studied because it relates to cancer very well. So a lot of money is going into this, into setting this up. But I think we probably got far enough for tonight.
Okay, all right.
Further, but it's interesting. When you say get to the bottom of it, I think we have entirely different ideas about what that means. When I can't speak for you, but when I say get to the bottom of something, I go down to the molecular nucleotide level and I look at exactly how it might have occurred.
Yeah, that's
So
the way.
I look
to
at
do it.
massive detail.
Yeah, that's the way to do it. I agree with you. That's the way I'm doing it as well.
Good. We agree on something. Okay.
Okay.
So I think I'll open this up if anybody else wants to come in. If you want to stick around and talk, I'm probably, have said as much as I'm gonna say for the time being. I will have more to say about you. I'll probably I apologize, but I will probably have more to say about this conversation when you're not around, or just individually. So I might do some more analysis on the whole thing.
Well, I mean, my resume here is, Mike, that you've come up with a diagram which I don't understand. Based on that, whatever you have said tonight, I cannot neither agree or disagree with you because I don't know what you are talking about. I would have to take you just on faith about what you said, which you know that I'm not very keen to do that. I expect that we would talk about logic gates, gene regulatory networks, which, since you are tired now, I've not been able to elucidate that further. But if you are keen to do that, we can do it another.
So what you're saying is that you don't have faith.
Well, why should I have faith, Mike, if I can investigate what we are talking about?
I don't know if you know, but the word Michael means God. I think it means like God. So
oh yeah, that's
I don't
nice.
know why you don't have faith in me.
Okay.
Oh, we got a bunch of people here. Okay, guys, I'm gonna mute for a while and shut the hell up. I'll let y'all talk.
Demeter,
Anyway,
so
thank you, Otangelo. That was
the
fun.
first thing I wanted to point
Okay.
out is that at the beginning you guys were talking about logical leaps. Do you guys know what a logical leap is?
It's an intuition.
No. A logical leap occurs when one has gaps in the logic of underlying their argument or their presupposition, what have you, and so they just jump the gap without filling it. A logical leap is essentially fallacious. You don't want to make logical leaps. It means that you're leaping in your logic because you don't have explanations for you're starting from a point and then jumping to the conclusion, leaping logically without actually connecting that premise to that conclusion. That is what a logical leap is. You don't want to do logical leaps at all. You don't want those. Those are not what you want in an argument. I also wanted to talk about epigenetic information. And Peter knows infinitely more than I do on this, so he is free to correct me anywhere I make mistakes. But as I understand it, epigenetic information is essentially contained in the chromatin marks in genes. So I guess one that I've heard is a methylation pattern where some of the cytosines in the DNA can be methylated, and that can form some form of pattern which can then essentially be the epigenetic information. I don't know how accurate that is, Demeter, but if any of that's inaccurate, please correct me.
So I will explain a little bit. Epigenetic could be very different in nature, meaning one type is direct methylation of DNA, which, as you mentioned, it's usually in the CPG islands. The CPG islands, probably approximately 95% of all of the genes before the promoter regions have a CPG island. And if this CPG island is methylated, it usually blocks the expression of the gene. So this is one direct into the DNA epigenetic code. The second one is the histone modifications, which is much more different. You have acetylation, you have methylation, you have butyration, you have many different modifications of the histones. However, what does the histones do? It just open or close the chromatin structure, or also attract the different regulation factors. So there is lots of regulation factors which recognize some of the some of those epigenetic modification of the histones. And of course, I involved again to express or block different genes.
But there are mechanisms for resetting that. I think on the gamete, and a lot of it. I mean, some of it is passed on 1 or 2 generations, And I think in the case of fungus, they found one that was 100 generations.
Yes,
I mean,
Jesus.
it's true. However, the nature of the different epigenetic modifications, exactly how long some of the epigenetic modifications are kind of more long-term modifications than the others.
yeah,
And also, in case of, for example, during the differentiation in normal conditions or in cancer, some of those epigenetics, of course, are remodeled. They could become different now because of the different regulations of the genes. And that's how it works. It's totally different, however, the post-translational modifications of the proteins. Post-translational modifications also a variety of it. The most common modifications or post-translational modifications are phosphorylation and dephosphorylations, which mostly are involved in a signaling pathway, as we see here, for example. Here you have most of the signals are simply phosphorylation or dephosphorylation of the proteins. And those are done by kinases, which phosphorylate, and phosphatases, which dephosphorylate the post-translational these proteins. So, and of course, they have some sort of specificity in order to phosphorylate or dephosphorylate. And now here's the trick. When you have phosphorylated protein, it changes the structure, meaning the actual 3D conformation of the protein, which might do either to activate or to block the activity of the certain protein. And this is why you have always kinase pathway in the signaling. So from the receptor molecule to the transmembrane receptor, so you have a hormone or some kind of signal which interacts with the receptor. And because of this interaction, you have conformational changes. And very frequently, uh, the receptor on the cytoplasmic side contains kinase domain, or could have domain which activate another kinase. There are many different variants, so I'm not going in details for this.
yeah. Cell differentiation depends on somatic line changes in epigenetic information that passes, when the cell divides, and then when you have the germ line. Okay, there are actually mechanisms that they found that resets a lot of the germline mechanism or the germline epigenetic information so that it does not get passed on. But then, of course, we have famously discovered stuff that does get passed on. But I think it would be like, for instance, it would be a horrible thing if all the epigenetic information was passed on, because then you wouldn't have stem cells. You wouldn't have cells that were capable of differentiating.
Exactly.
Right.
This is not so. I mean, the stem cells, by definition, are cells which are not differentiated cells and
Right.
to renew itself. But I wanted to point something else here. If you go back to the diagram, which Otangelo showed us,
Oh, the mTOR?
the same pathway that we just looked.
Yeah, the mTOR pathway. This is my favorite pathway, by the way, because it shows spaghetti monster very clearly.
Yeah. I mean, we studied that, and I actually personally was working with some of those. So first of all, I want to mention this is a kind of diagram which is principal diagram, meaning it's not presented everywhere every time. Some cells have some pathway, and other cells have different pathways.
Okay.
So you should not confuse that everything is in one cell.
Right.
And that's one thing. The second thing, if you see the bottom left, on the left side, you see the structure of the protein mTOR here. See that small?
Yes.
So, and you see the protein from left side to the right side, and you see the different blocks, small blocks here. So
the heat repeats.
Heat repeats, heat repeats, FAT, FRB, kinase domain, fatty C. See that?
Yep.
So
Yes.
this is the domain structure of the protein. So every protein have a domain structures. Now, the something which everyone should know that this domain structure it's actually reducible. Meaning, if you take kinase domain, which is on the right, the green small bar here.
Yep.
So if you see this kinase domain and you attach this kinase domain to another protein, the other protein will become kinase. What is the kinase domain? The kinase domain actually do phosphorylation.
Yep.
It just need to fit and to do the phosphorylation event. So this is how actually those proteins evolve, signaling pathways. It just by translocation, amplification and translocation, you can have new protein which contains different combination of the same domains that you see here. And if you see the same domain is represented for the interaction, it's responsible for interaction of different with the different signaling molecules. Like, for example, you can change the interaction pattern in this way. So even theoretically, you can create totally new protein just by the combination of those domains. And actually, that's what happened in cancer. Very frequently, you have recombination of those domains, and you can have so-called fused proteins. And some of the cancers are very well proven to become from de novo protein, which is a fused protein, like early sarcoma, for example, and many other. Also, of course, if you delete, so very frequently you can see P53 here, the famous P53 here. The P53 is a tumor suppressor gene.
Yeah.
So, in P53 is also a transcription factor. And of course, if it happened that it's mutated or deleted, all the other factors will take over and will push the cell toward proliferation. So the evolution goes it's explainable very well here. Just by rearranging these domains and making new proteins. That's how actually these pathways occur. And it depends on the conditions. In fact, you can create simply another possible protein which it's happened to be suitable for the particular purpose.
Yep.
And that's how it works.
So I think it's really easy to see how evolution by mutation happens by looking at this type of mechanism, this type of complexity. I don't know.
I mean, yeah, sometimes point mutation might be devastating, but sometimes point mutation might do absolutely nothing. So,
yeah, it might reduce the pathway, or it may put another step in the pathway, or or modify, create another inhibitor or enhancer or whatever. But I mean, it's just obvious to me. Evolution is so obvious when you look at something like this.
Also, the entire pathway here, it's reducible. It is reducible in a way that you can delete some of those factors.
Yep.
And the cell will still viable, and even sometimes might not have a significant phenotype, or it depends on the situation, might just arise a cancer, or it depends on where, in which cells it happened. Could be either proliferative or not. It depends on many other factors. So, okay, that's what I wanted to say.
Yep.
Any questions about that?
No, I don't have any.
Well, I believe that I've developed some additional pathways that make me smarter. But that's just my opinion. Anyway. Okay. Is Immutable still with us? What the hell happened to him?
No. Who the fuck knows? That guy's probably out walking around like a
He's
like
probably
a fucking
walking.
troglodyte.
I think he goes out to meet his drug dealer or something, or like I don't know, his girlfriend. I don't know what it is. I hunts alligators. Who knows what he does down there in Florida? Anyway,
I'm gonna capture me an
Okay.
alligator. I'm gonna stick my thumb in his butt.
yeah, yep.
Also about this, how was the logic, immutable logic?
The what? What now?
The logic that it's how it's the name
Boolean logic.
relatic logic,
I'm not sure.
Otangelo.
Yes.
Otangelo?
How you call this logic? I just the word escaped from me.
I apologize.
I think
I was searching here information where the glycan code is stored. Science paper.
Immutable logic. This
I think it was Boolean logic.
Boolean. Okay, sorry about it.
Boolean logic. Yeah.
Boolean logic. So
Yeah, it's not very good Boolean logic
in,
because
in
it's analog.
biology, there is no clear Boolean logic. It's not sharp like yes or no. However,
Yeah.
you might find some cases, of course, but it's like a 1 or 0. You know,
Yeah.
yes or no or maybe. Or it's this. I've studied this logic. I simply didn't know that it's called Boolean logic.
Well, there's a kind of electronic circuit.
Yes.
It's called a latch. That basically, if you give it a little tiny signal, it'll feed back into itself until it becomes a very big signal, and it'll latch one way or the other. So it'll drive to 1 or 0. But I mean, in biology, we have in between ones and
So
zeros.
because I personally
have a
studied
question.
some of these pathways here that you can see here. I mean, I prefer much other pathways I've studied, not exactly mTOR pathway, but it's still, there are lots of
I have a,
common. But
I have a
my point
I have a
is,
question.
just to finish that sentence, it's never, these signaling pathways are not like a yes or no. They are
Yeah.
more like graduated. So you have stronger signal and less and weaker signal. So you may have very strong signal or even weaker signal. And that's definitely not like a 0 and 1.
Yep.
This is all of the signals that you see are not like yes or no. They always, in fact, we always have, when we did a study, you always have some sort of problems in terms of to block completely some pathway.
Yeah.
Most of the time you just go down to the signal pathway to some level, but you almost never succeed to completely
wow.
block the pathway. And that's happened all the time in our research. The second thing I want to mention, not just even that it's not like a yes or no, but in development, sometimes the development pathway, like a splitting between mesenchymal epithelial cells, for example, it depends on the gradient of the signaling pathway. So again, it's not yes or no. It's not 1 or 0. You have
Yeah, like
you have a graduation, and because of this graduation, for example, BMP pathway, it's very clear as a graduation in the very beginning of the embryo. It makes a difference between epithelial, mesodermal cells and endodermal cells.
yeah,
I
the
still have a question.
hang on a second. Basically, the zygote from the posterior to the anterior, there's like bicoid and caudal on the posterior, and it's a gradient between those that develop all of the initial segmentation, like in a Drosophila, in a fruit fly, and the gradient is it's really interesting how like 40% will kick off one particular pathway and 30% will kick off another, and it forms into like 10 segments along the posterior anterior axis. Anyway, okay, Leo's turn.
Otangelo, forget about Boolean logic.
So what is
Now,
this?
Mike,
What is this?
Oh,
Hold on,
hang on a
because
second.
I've been trying to ask a question for like 2
Leo's
minutes
been
here
trying
now.
to ask you. Yeah.
What is this boogeyman logic you guys keep talking about?
Boogeyman logic?
What is that? Yeah, that you guys keep mentioning. What is boogeyman logic? What is that? I
Bully,
don't know what that is.
Bully, and George Bull. Oh my God.
I
Are you
got
playing
to keep.
with us? Leo's playing with us.
Is Immutable dead, or is he just... Did he
I
lose
don't
his
know.
voice?
I think he's dead. He's probably just
Okay,
past the
guys.
black.
So see you tomorrow.
Okay. Well. Oh yes. Okay. Are we doing your question and answer thing at some point, Demeter?
Oh, I lost any hope for that. Anyway, so it doesn't matter.
Yeah. Let's try to do something this weekend for sure.
It's up to you guys.
Okay.
So
See you.
I'll be around, but
Okay.
mostly
Is...
in the morning until probably 5-6 o'clock.
Okay. So morning. Like, how do you define morning?
I'm defining everything between 10 o'clock and let me say 3 o'clock.
10 o'clock is not morning. That's afternoon for me.
For me, it's boring.
Okay, we'll try to hit that. We'll try to set up maybe like a Q&A thing on your papers.
Could be.
Okay. Well, we'll do something.
Somebody,
We promise
somebody
we will
needs
do
to,
something.
Somebody needs to shake a mutiny. Or table desk, whichever one you prefer.
A mutiny. Table desk.
What's going on?
Okay, he's there. He's alive.
Yep.
He is alive. Well, contribute, damn it, because I'm done contributing.
Okay,
What-
Mike, I just would like to respond to you. You said that the glycan code is stored in the genes, and I've investigated your previous chapters have described how glycans are not directly encoded by genes, and that they are biosynthetically and structurally complex.
So they come from the zygote, basically, in development, is what you're saying.
Well, what I am saying is that they are the glycans are not encoded by genes. That's what
Where
that
do they come
paper
from?
here confirms.
So
Well, that's what I am
Ingo,
saying. That's what I am saying. That the structure is there is in the the sugar attached to the protein. Now, where that information comes from, that is the big crux.
Okay,
Well, that
the
comes
big
from
question.
chemistry.
Let's
What?
explain.
Wait, what are you guys talking about?
Glycans. Demeter, do you
How
want
do
to
you spell
respond?
that?
I want to explain.
How do you
this.
spell that?
I'll put it in chat.
GLYCANS.
Glycosylation.
GLYCANS?
Yes.
Okay.
So let me explain. So this is a post-translational modification, and lots of proteins actually go through glycosylation. In order to go that, they first after the synthesis of the protein, they go through the Golgi apparatus in the cell. And
Yeah.
Golgi apparatus, there is enzymes. Actually, those enzymes are set up in a particular order. And these enzymes are glycosylases. So I don't know exactly details here because it's not in my field, but I can explain simple as that. So during the going and pass through the Golgi apparatus, the proteins receive this glycol chains or link with, and actually it's like a code because it's a branching different sugars, different types of sugars. For example,
Yes, correct.
funerose, let me say glucose, fructose, and those, and they have a different links, alpha, beta, 1,
What the
2, 1, 6, 1, 4, and
Dude,
those
go the
kinds.
fuck
And
away.
you
What
have
the hell?
like a tree of glycolytic tree over the protein.
Yeah, it's like a tree structure which
Yeah.
peers information.
And this
Right. It'd be very, very, very complex.
it
and
is
has-
complex, yes. And also it depends of many other things. For example, which tissue. And those proteins mostly are made to go in the membrane. And most of the time
Yes,
just
yes.
stay on the membrane in between the cells in the cell matrix.
Exactly.
But how, okay, Demeter, how are the glycans manufactured? How are they created? They don't arrive from
So
space.
it is every single branch and every single adding of the sugar comes from the particular enzyme.
Yeah.
Yep.
So the answer directly from the Otangelo's question, it is coded but it's coded by enzyme. So the code for this glycolization pathways comes from the enzymes, meaning they are not directly coded, but they are built up from already coded enzymes. So you still have a coded enzymes which comes from the
DNA.
DNA.
Right. And that's what I was trying to explain to Otangelo.
Yeah.
They don't just. I mean, just because we talk about the glycan code because we found the complexity of glycans to be amazing, doesn't
It is
mean
code.
that.
Yes, it is. It is very. It is really complicated. It's not too simple to understand, and also it's very difficult to study. By the way.
Well, I am not sure. It is possible that partially the information comes from genes, but it is known that the glycan code is far more complex than
Yep.
only genetic information.
Yeah.
And the reason for that is because glycosidic linkages can be two types. So you can have O-glycosidic linkages and N-glycosidic linkages. And the majority of sugars that are present in a cell are either 5-membered rings or 6-membered rings, which means typically you have 4 different types of bonds that can be made with the hydroxyl, with different hydroxyl groups based on where it attaches. And you can have 2 for each of those based upon whether or not the orientation of that bond is alpha or beta. So it's because of the internal structure of the glycan that makes it more robust.
Boogeyman logic. Yes, and that just shows that life is complex.
So Otangelo, just because something is complicated and very complicated, it's not the same as being intelligently designed.
Well, Dimitar, that is strawmanning what I am saying. I am not making that argument.
Okay.
Logical leap.
Okay. So what else I can say? There's something else I would say. Sometimes the things sounds very logical. I agree with you. But see, it's also true that not every logical thing that sounds logically are correct.
Yeah.
You have to understand that as well. And
Well, I mean, if someone has, I mean, I think it was Dawkins that said that biological systems appear to be designed, but they are actually not designed. And in order for him to claim that, he must substantiate it. And of course, you can say what appears logical is not logical, but when you say that, then you must substantiate why something which appears logical isn't logical or isn't what it appears to
I
be.
agree with that. Yeah, that's it.
And that's kind of what the purpose of Dawkins' books was to do. That
Yes.
from like the first page of his book.
So let me be clear. Everything is logical. After all, everything is logical. Simply, I want to say that because something appears in front of us to be illogical doesn't mean this is the correct logic.
Well, maybe logical is not the appropriate word. I would say something appears to be made in some sort of way, but eventually it isn't actually that kind of way which appears to be.
Exactly.
That
Yeah,
is,
I agree
well,
with
that,
that.
well, that is possible, of course, but when you say that, you must substantiate that and say it's not as it appears to be because of this and that.
Yes, I agree with that. So, but obviously that's a different level of discussion. And just shortly, very shortly, without going in details now, but I can say, yes, if you see how and logic about how the things and the mechanisms are evolving, you have explanation. So that's it, simple as that. I mean, I can give you back your previous example with the car. You remember the car in James Store example. Remember this car?
Yes, of course.
So the car which is sitting on the beach, disassembled in the parts. So in that case, for example, you can see a wrong model. For example, it's never been the case that we said that the thing started this way, the steady state way. It's simply you should not present the wrong model in front of the people. Also, different things about the car. You go and look and see the car, right? And say, oh, this is artificially created object, intelligently created object, right?
Yes.
And we agree, all of us agree that if you see a car, you can say, yes, it's a very intelligent design, especially if it's a Volvo, for example. uh, some are not so intelligent created cars, but that's a different level of discussion. So why we say that? Because there is no way to say how naturally the car occurs. We don't have clue how this car could appear naturally. You don't see a law, you don't see how naturally could occur. However, if you see a cell and say, oh, that's a very intelligent design, yeah, it's appeared, but you now start to see, when you start to study the cell, you see all of these domains. You have phylogeny, you have mutations, you have all mechanisms presented in front of you, and how those could happen naturally. Therefore, you cannot go and conclude, oh, that was intelligently designed. Let me say it this way, it is designed very intelligently, but by selection, naturally it's designed. So,
I can do.
okay, if everybody is tired,
I'm tired.
I have to go.
I'm never
I
tired.
can draw boxes around my picture. Did you see that? Look at that. I can draw boxes around it.
Oh, yeah, that's nice.
What?
Yeah, I didn't know I could do that. I wish
I
it
don't
were
see
brighter.
anything.
But anyway,
What box?
Okay.
I don't see a box.
Immutable, you're tired?
Yeah, man.
Yeah, you're going to go on some
Always tired.
stream for about 5 hours tonight. So
Oh yeah, definitely. That's
Yeah,
exactly
you
what's gonna happen.
I just hauled in a gigantic 5-pound plate of
I was up
Chinese
until
food. What?
I was in Praise's stream last night, and from like
I saw
until
that.
4:30 in the morning. I can't remember when I hopped in. Yeah. Oh, you were there?
Well, I popped it a few times, but how many times did
got-
Gavin mute you?
Tap it.
How many? You're roboting. No, you're frozen.
He muted me and Mr. Parker literally. Well, me more so dozens of times because you know how that's how those theists work. They can't.
Yep.
Okay, guys, I think I will leave the stream.
Thank you, Otangelo.
Thank you for stopping by.
Okay, thank you, guys. See you next time. Bye.
Okay.
Yeah.
God, this is so fucking stupid.
I think I'm going to leave the stream. You guys want to keep going, or
No, I'm done.
you're
I'd
done?
say goodbye, but my internet is so fucking shitty because I live in rural Iowa.
Yeah.
Well,
Yeah,
I'd
the,
say yes, but nobody can hear me because I'm fucking frozen and roboting, so it doesn't even
Yeah.
matter to comment at this point.
I used to live in rural Minnesota, or a place called New Germany, about 50 miles west of the cities. And we had like a 4600 baud modem that you had to put a phone in the beginning, and that kind of sucked. And they haven't come a long way since then. We really need internet, like satellite internet that covers the entire United States, but because it sucks. Anyway, guys, so I'm going to end the live stream. So goodbye, Icarus, and hi, Randolph. You just got here. Now
Am
we're
I
leaving.
back?
Oh, you're back now, yeah. Yep.
Am I back? So wait,
are- back.
how much of it did you hear?
Nothing.
Yeah, no, how much of it did you hear?
Oh, last night?
Well, no,
I
not
just-
what I said last night.
Last night. I just stopped in for a little bit here and there, and then I fell asleep. I was watching a movie, so I just kept checking on you because I was worried about you.
So did you miss that every time I would press John to answer, and I'm calling him a Yanni boy now. That's the new.
Oh, Yanni boy.
I'm gonna start calling
Yanni
him.
boy.
Every time I would try to press him on a question, he would run to Gavin to mute me because. They're just they're just intellectual cowards. There's no other way for me to put it. They're just intellectual cowards. Straightforwardly, intellectual cowards. And then Jungle Jargon tried to educate me on special relativity, talking about like length contraction and time dilation. I asked him literally probably 30 or 40 times to give the equations for him, and he couldn't do it. I asked him to give equations for practically everything he said, and he couldn't do it. I asked him what Lorentz transformations are. I asked him what a Lorentz boost is. I asked him to define the difference between special relativity and general relativity, which is a trick question because they're the same thing, just defined on two different manifolds. Let's see, what other questions did I ask him? Holy shit. I asked him what a complex number was. I asked him if he knew anything about calculus. I asked him if he knew what a Minkowski spacetime is. I asked him if he knew what a pseudo-Riemannian manifold is. I asked him if he knew what a Neo-Lorentzian manifold is. I asked him a lot of questions related to what we were talking about and didn't answer a single one. And then every time I pressed him to answer them, they just, well, he was just like, oh, you need to mute him. And I'm just like, wait. This makes no sense. I'm asking you questions. I've answered all of your questions. You haven't answered a single one I've asked, but I'm the one that needs to be muted. And eventually, towards the end of it, the one other Christian there actually took my side and was like, yeah, Jungle, I think that you really don't understand what you're talking about.
What other Christian was that?
I can't remember whatever the fuck his name was. I think he told me to call him Will.
So was Maddox on the whole time?
For a little bit. He was there for a good while. And we jumped around from topic to topic because every time I kept pressing them on a topic that they couldn't, provide cogent responses to, they'd hop to something else because, that's what they do. We were started on morality. And that was really fun because I tried to, what I tried to do was pin them down on, if we get our morality from God, which is what they believe, and we think rape is wrong, then God thinks rape is wrong. If we would act on that morality to stop a rape, then it would necessarily follow that God would do the same. So then why isn't God stopping rapes? And then praise worded it as, well, that's raping the other person's free will, which doesn't even make sense. And so I asked them, wait, so is letting a rape happen because you don't want to intervene on the free will of the person doing it
I heard that
more moral than stopping the rape? Because if it is, then you just don't have a good sense of morality, and obviously neither does God.
Yeah, I did listen to that part as I was falling asleep. Where did they go with that? It was awful.
They didn't go anywhere because I kept trying to pin them down on their own worldview, and they couldn't do it because they knew what the consequences of it were. So then
they said that,
we jumped to something different.
Well, didn't they say something like, if you make something or you create something, you don't micromanage it? They went to God is not a micromanager. I'm pretty sure somebody said that, and that was where I pretty much fell asleep. I thought it was funny.
Okay, but if that's the case, what the fuck was the flood?
Yeah, exactly. Other than micromanaging, and and what happens when you pray for?
There are numerous examples we can use about where God obviously has a relation to morality.
When you pray for your cancer, what is that called? Is that micromanaging? I don't know. Anyway, cool. I am going to check this out a little more. I'm interested. Did you guys cover evolution?
I wanted to jump about something I said.
Oh dear.
Oh, you're botting again. Oh my. Where's
No. Well, Standing was in there for a little bit. Was asking me questions that weren't really questions. So, I mean, what? Give me just 1 second and try to figure out why the fuck this keeps roboting.
There's an echo coming from somebody's mic. I think somebody's replaying the stream, maybe?
Yeah, somebody is echoing in the background real quietly. And I don't think it's... Oh, maybe it's me. It ain't me, I'm muted.
Huh.
So no, not me. I don't know, I can mute and see.
I don't think it's me because I still hear it after I mute myself.
Maybe it's Leo.
Leo, I think you're playing the stream in the background. Leo?
Me?
Yeah,
No.
I think you're echoing. Try muting.
No, I'm not echoing. It's
Try
definitely
muting.
not me echoing. I don't echo.
Try mute.
Why? I've literally never echoed before. It's probably my TV. Do you want me to turn my TV down?
No, I actually hear myself.
Well, it's not from me.
echoing in the background.
It can't be from me. I have a headset on. The mic can't pick up what's coming through these.
It is me. I don't
I don't
know
hear
how.
an echo.
I do.
But so no, but you asked me before I started roboting in because StreamYard is just shit. So Standing was in there and he was asking questions and I could tell they were non-questions. That you know, you guys know what I mean when I say non-questions because it's Standing. We all know that's what he does. He asks questions that aren't really questions. And he'd ask something and I'd be like, well, I can't answer that question. There are people I'd have to talk to. I'd have to talk to some professionals. And I could tell he didn't really know how to respond to that. Like he would just be like, I've got these papers, and it's like, cool. I would need to take these papers, go to a professional, talk with them about what it says, and actually dissect the minutia of what the paper is saying. I don't know what you want. Because he's not used to that. He's not used to somebody actually admitting their own ignorance on things. He's used to people either thinking they know what they're talking about, or I should say people that actually know what they're talking about, just refuting his bullshit, or getting somebody who thinks they know what they're talking about and giving non-responses to his non-questions.
Standing and intellectual honesty are a very complicated pair.
Yes. And Maddox tried to tell me that simulation theory is a viable theory, and I'm like, it's not even a theory.
No,
I got
it's
him
not.
to rage. I got him to rage quit. He tried to tell me like, so wait, like gravitons and string theory, those aren't theories, and I'm like, no, they're not at all. They're not verified.
they're not
One of the
theories.
marks of a theory is, well, yeah, but mathematical theories aren't scientific theories, and what he was referring to was a scientific theory. Something that is well substantiated, repeatedly tested and confirmed explanation of an observed phenomenon. And we've never observed gravitons because they probably don't exist. But that's a completely different discussion. Yeah, I got him to rage quit.
Who rage quit?
I got John Maddox to rage quit, and then he came back in like 10 minutes later.
I'm gonna bail. I think I'm the one that's echoing. I have no idea why, but
I don't hear
I'm gonna
an echo.
bail up.
Why
I
don't you
do.
refresh your mic?
What's that?
Refresh your system.
Yeah.
If I refresh StreamYard, you mean?
Yes.
What'll happen if I do that?
Probably the world will end. I don't fucking know.
Dude, I am so sick of this bullshit.
How about now? Yep, still like.
Why?
Anyway, I'm gonna go.
Dude.
Do you guys want to end the stream, or do you want to continue without me?
I guess it depends on what everybody else wants.
Yeah, I'm okay with whatever.
I'm willing to stay in longer, but I just depends on what everybody else wants to do. What the fuck just happened?
I think we're bailing. I think we'll end the stream. So I'm gonna. What's
what
that?
the fuck is this?
We don't know, Leo.
What the
We'll figure it out
shit?
after we end the stream.
No. I'm talking about the MDD between Matt Dillahunty and Stuart Neckley. It just.
Oh.
What the shit just happened? They must have been having technical difficulties.
Anyway. Okay, guys. Goodbye, everybody out there. I'm ending the broadcast.